Okay, our next speaker is Jae Kim, and he's going to give a talk on novel behavioral paradigm of interval timing and Drosophila.
Thank you, Jacob, and thank you, organizers, to giving me a chance to present my work. So this work will be just introduce you about the new paradigm. So it's a lot of data, so there will be not that many details of the data. So there are several issues about timing. So as the Amical Siegel and the KISS said, there's a circadian rhythm, and the sleep is called the circadian timing, like a 24-hour, 12-hour cycle. And also, there are millisecond timing, something like a very short time we can measure. But there are, in my knowledge, there are no known behavior that can mimic the interval timing, that is minute level measurement. So what is interval timing? So interval timing allows us to subjectively experience the passage of physical time. So why is it important? Because this allows us to integrate action sequences, thoughts, and behavior, and to detect emerging trends and anticipate the future outcomes. So interval timing in human being is very important cognitive abilities. So there are theoretical interval timing model from 1960s. So there are several components to measure the interval timing. First one is a pacemaker that emit pulse that are gated into accumulator by the mode switch. We call it clock and perception stage. And next stage, the working memory is compared with the previously stored reference memory for that particular event durations. So then you can compare how many minutes you will decide to perform some actions. So this is a comparator. We call it decision stage. This was suggested from 1964 from a church, but no known human circuit is yet to be determined. So my question is that can we make some model in fruit fly that mimic this human interval timing? So what would be the mode switch for flies? So what I found in GenLab is that when males expose the other males, we call it livals, sexual livals, they extend the mating duration significantly, about 5 to 7 minutes. The flies live like 60 days. So 5 minutes for flies is like 2 months. So they sex like 2 months more because they expose the lival. It's a lot of effort for investigation for flies. So we call it longer mating duration. So I named it. So it helps to increase the fitness of males. And it published in 2009 from the UK group that they called a very complex name, but I rename it as a longer mating duration. So what would be the sensory input for rival? That fly, how can they recognize the rivals? So I tested some mutants, like olfactory mutants, auditory, gustatory, and visual mutant. So as you can see, whenever you use some visual mutant that is blind males, they cannot perform the LMD behavior. But this is a mutant, so there must be some simple way to prove that visual stimuli is necessary and sufficient to induce LMD behavior. So there's a simple way. You can show them a mirror. So I showed a mirror to the single-eared fly, and I found that they made longer. So they think this object is a lival. So I don't think they think that that's themselves. So I proved that LMD is solely induced by the visual stimuli. So we found that there are mode switch in fly that can induce the interval timing. So where is the pacemaker neurons and where is the accumulator neurons? So I tried to do some genetics. So there are, thanks to the chronological biologist, we know that this clock cycle period timeless feedback is regulate the circadian rhythm. So I tested all the mutants of the flies, and surprisingly, what I found is when the period and time is mutated, the LMD just disappeared. But the clock and cycle mutants still show the LMD. That means the clock and cycle function and the period of timeless function is separated in terms of LMD, not in circadian rhythm. So, but there are several behaviors that is very similar with this. So, may co-shift some frequency, development timing, and sleep length, or some cocaine sensitizations, clock and cycle, period and timeless function can be separated. And so, where is the clock neurons? There are 150 clock neurons in the fly brain from the 100K neurons. So it's really well mapped. So what you can do is neurocircuit mapping with the genetic tools. So I will not explain this. So I try these gophers with the KL, with the tubulin RATS to drive KL only in adult-specific manner. And I also try to rescue period mutation rescue experiment with these gophers. So this is a very complex story, but make it simple. What I found is period function is only required only these 8 neurons. That is necessary and sufficient to induce LMD. And the neural activity is required in these neurons. That's what I found. So I was asked the question, how they communicate each other, these neurons. So I tried to screen the neuropeptide, how they, that connect these neurons. So there are 3 major neuropeptides in fly clock neurons called MPF and PDF and small MPF. So I briefly tested the mutants and found out that only PDF and MPF mutant show the defects in LMD, not SMPF. That means SMPF is not involved at all with the LMD behavior. So are all these neuropeptide expressing neurons are required to show the LMD? That was my question. So what I found is it's not. Only these 4 neurons expressing PDF is necessary and sufficient to show the LMD. So large LMD 4 neurons are not required at all to show the LMD. I mean the PDF expression there. And the 2 MPF and male-specific neurons are necessary and sufficient. The MPF expression there is necessary and sufficient to show the LMD. That's what I found. So this is neuropeptide. So where is the neuropeptide receptors that get the signal from there? So this is my strategy. So I collect the gophers expressing in different subsets of clock neurons and knock down PDF receptor or MPF receptor to different lines of RNAi and test the expression pattern of those gophers that I see the defects in behavior and narrowing down with the GAL80s or narrowing down using different gophers and did the rescue experiment with the gopher GAL80 combinations. So I will not show you all the data, but what I found is this. The 4 neurons in here, sorry, this doesn't work. So 4 neurons here, the PDF expression signal go to these 2 neurons, PDF receptor expressing neurons. And there are 2 neurons that express MPF, then male specific. They send the signal back to this neuron because MPF receptor is expressed here. So there are feedback group that is very important to calculate whether the libel information in fly brain. So maybe we found that pacemaker and accumulator in fly brain that show the interval timing. And where is the reference memory and the working memory for this libel information? So what I found is, this is just summary. So what I found is LMD disappears between 6 and 12 hours after libel exposure. That means that is long-term memory. And there are classical memory mutant in fly, Rutabaga, amnesiac, DUNS. So only Rutabaga and amnesiac is involved with LMD behavior. And There are several brain regions that is very important for fly memory. So I test several brain region Gauss force to drive the KL, and what I found is ellipsoid body neurons are very important for visual memory of these levels. And what I found is root vega expression only in R2, RM4 neuron is necessary and sufficient to restore the LMD defects. So maybe we found that where is the memory is working in this interval timing model of the flies. So where is the comparator? This is very important neurons. So in myself, I cannot find it, but the Michael Crickmore in Harvard published this Cell paper 2013. And he blocked the function of the corazonin neuron in abdominal ganglion. There's 8 neurons. Then what he found is fly made 2 hours if you block that neuron function. And he did very interesting experiment that he decapitated male brain and measured the mating duration of those 2 different flies. And what he found is, he found the differences between decapitated flies and the normal flies. That means there are some connection between this corazonin neuron and the neurons that I found. But he didn't explain in this paper, and we will discuss about that. So I suspect that might be the comparator neurons. There are no functional data yet, so maybe corazonin and GABAergic signaling may be very important for these comparator neurons. So I think I found some very interesting behavior paradigm that can we study the interval timing that is very, very important for human cognition. So this is the summary of the fly model of interval timing. So LIBO signal is just caught by fly male, by the visual stimuli, and this signal goes to the PDF MPF signaling pathway. Then this is the memory circuit for this information stored in the ellipsoid body. And the comparator neurons are in the abdominal ganglion, that is corazonin positive and GABAergic neurons. That comparator neurons then fly decide, I will make longer. So there are another mode switch, I think. This is the paradigm we are working on in our new lab. That is we call shorter mating duration. So if you have any question about this interesting behavior, just let me know. Just ask me a question later. So there are several things, because my lab just started last year. So there are several interesting questions from the input and the output. So we are searching for their specific sensory neuron and specific motor neuron for this behavior. And we also try to find, using the neuropeptide and neuropeptide receptor signaling, we want to find the intermediate circuit that regulate these very specific behaviors. Okay, so poster session ended, but my first master student had a poster, maybe you see, and there were very interesting posters by my tech about how you make a cost-effective lab, because we are saving like $20,000 per year by this method. So if you have a question, just send me email. I will let you know how you can save your money. So this is my lab, and thank you.
We have time for one question.
I'll just ask, does the female have no say on how long the male does his interval for copulation? She'll just sit there for 2 hours with a male hanging on?
No. The average time for male mating duration is 20 minutes.
No, but the mutant, I mean, clearly if the male is sitting there for 2 hours, the
Michael's thing? Yeah. Over 2 hours.
So clearly the female's not shaking him off in disgust.
Mating duration is solely driven by male.
Yeah, that's what I asked. Huh?
So what female do is, until the male mount, female can decide whether reject or accept. But after that, female do nothing. They just eat and just let the male stop the mating.
Huh.
So that's the natural behavior of flies. Yeah.
I have one quick question. So how long, how does the long mating duration depend on the length of time the rival is there? Is it just Does the rival have to be there the whole time?
Yeah, so exposure to libel, at least you should expose to libel like 12 hours. So there are some threshold. The threshold should be over.
But the rival doesn't need to be there at the time
Yeah,
of the actual
the present libel doesn't matter for this behavior. So you already know. Yeah, I replicate that.
Thank you.